give it just a minute and a couple other people log in. Okay. We left off talking about proteins and separating them into primary, secondary, tertiary, and quaternary structures. So these things get layered on top of each other. So the simplest structure is a primary structure, and this is the peptide bond that we talked about. So taking two amino acids and putting them together, we kind of think of the amino acids as pearls, and the primary structure is the string that chains the pearls together. So a peptide bond is an example of a primary structure. If we have two peptide bonds, dipeptide, 3, tripeptide, 4, tetrapeptide. If we have a bunch of these guys lined up together, we call it a polypeptide. It's an amide bond, so we create a bond between the Cu group on one and the amine group on the other. We can take these two separate guys and chain them together with this peptide bond. This was where we left off. Hold on 1 second. Yeah, okay. So we talked about when these get named. So with the exception of the C terminal, so the one that has its C group sticking out, that we just use the full name. All the other ones will get YL'd at the end. And then we just stick them together. You can also shorten it using the three letter abbreviation or the single letter abbreviation. I believe we talked about this and how we would do an abbreviation for this. ALA, LEU, CS, CYS, MET. So a protein, what we classify as a protein, which is a really large molecule, is a polypeptide of 50 or more amino acids that has some sort of biological activity. The primary structure of a protein, so the most basic structure of a protein, is going to be amino acids held together by polypeptide bonds. So again, these structures, the secondary and the tertiary structures will get layered up on top of these. So our basic structure is this polypeptide chain. So here we can see this is one amino acid and it's chained together with our polypeptide bond. Here's our next amino acid chained together, next amino acid, so on and so on. We can see our R groups are sort of sticking off the end. So the thyroid hormone that stimulates the release of thyroxine is a tripeptide. And this is its amino acid. You don't have to have that memorized, but right, when you start to hear things like tripeptides, dipeptides, this is what's happening. There are other amino acid sequences with these three but this specific sequence is what gives us the proper hormonal activity. We can see insulin over here. This was the first protein to have its primary structure determined. So they were able to take insulin and they were able to sequence its amino acids. What was the correct ordering of its amino acids? There are two chains here. We have an A chain, which has 21, and a B chain, which has 30 amino acids. So the B chain is a little bit longer. Its primary structure are 2 polypeptide chains, and these are linked together by these disulfide bonds. And we'll talk about these being what kind of structure this is here. But the primary chain is this here, and this here we would classify that as a primary structure. So it says, answer the questions for this tripeptide. What is the N-terminal amino acid? So I wouldn't expect you to have the amino acids memorized, but the N-terminal is this over here where the N is. The C-terminal is this over here. And then if we were to name it, this would be the name. I'm not expecting you to have the names memorized of the amino acids. It says, write the names and 3-letter abbreviations of the tripeptide that could form from 2 glycines and 1 alanine. So as far as the different possibilities, if you have 2 glycines and 1 alanine, you could have a glycine, and an alanine. You could have a glycine, an alanine, and a glycine, and you could have an alanine, a glycine, and a glycine. All right, so either the 2 glycines clump together in the beginning, the 2 glycines clump together at the end, or the glycines split. All right, so that's all the different combinations. And this one here and this one here are different from each other, even though the glycines are grouped together. Here, the glycine will have its N group exposed. Here, the glycine will have its C terminal exposed. So let's talk about these secondary, tertiary, and quaternary structures. So this staircase here looks like a coiled DNA, right? The shape of an alpha helix for DNA is what's known as a secondary. It's a type of secondary structure. The alpha helix acquires this coiled shape from hydrogen bonds that are happening within the structure. So let's look at some different type of secondary structures. So if we have an alpha helix in a secondary structure like the alpha helix, there are hydrogen bonds forming between adjacent oxygens and hydrogens from, here's the oxygen, you can see here, there's an oxygen from the C group on one end and the hydrogen from a C group on another. And what that does is it causes it to coil in on itself. So you can kind of think of the secondary structure as, like if you remember, anyone on here is old enough to remember old school telephone cords, right? That coiled on themselves. So you had the cord, which if it was really old and it was long and stretched out, right? It didn't have any secondary structure. It just had the primary structure. So you kind of had the pearls linked together. The secondary structure is when it curls in around itself. So we get this by the formation of a lot of hydrogen bonds along the peptide chain that will give it this spiral staircase shape. You can see it here. And remember, this hydrogen bonding is slightly weaker than the bonding that's actually making up the primary structure. There's another type of secondary structure. This is called a pleated sheet structure. So in this secondary structure, we have, sometimes it's called a baited pleat sheet. We have hydrogen bonding forming between the carbonyl oxygen atom and the hydrogen atom in the amide group, and it bends to form a sheet, kind of like an accordion. And would I expect you to draw this structure? No, no way. But I do expect you to know some of the definitions of this. So there's secondary structures, there's an alpha helix structure, there's a beta pleated structure. Both of these involve hydrogen bonding, things like that. Here is another secondary structure. This is called a triple helix, so different than the alpha helix. In a triple helix, you have 3 polypeptide chains that are woven together. Like if you were to sort of braid your hair, you have hydrogen bonds holding the 3 chains together, giving the polypeptide strength. And this is something that we see in collagen, connective tissue, skin, tendons, cartilage. As we get older, this beautifully tight triple helix starts to sag a little bit. That's why we lose some of our collagen and some of our connective tissue. Our skin starts to age on us because this doesn't have quite a strong bond to it. So we have our primary structure, peptide bonds. We have our secondary structures, which can be alpha, beta, alpha helix, beta pleated, or this triple helix. Now we can layer this into a tertiary structure. So this tertiary being for 3, the 3rd level of structuring. The tertiary structure of a protein is an overall 3-dimensional shape caused by interactions of different parts of the chain, causing it to bend or twist. So it is determined by cross links, or this is the attraction or repulsion between the side chains or the R groups of the amino acids and the peptide bonds. So looking at, I'm going to go back to this here. Let's say that this is our chain. So our single chain would be, or our primary structure would be this peptide bond connecting the amino acids. Our secondary would be this O interacting with this H to form a helix or a pleat. That would give us a secondary structure. A tertiary structure is these big accessories that make the amino acid, its characteristic amino acid. Those things start to interact with each other. And they make the tertiary structure, the string, whatever it is, if it's an alpha helix or a pleated or this triple helix, it makes it fold in on itself. So think about taking your phone cord that's curly and then folding the phone cord in on itself because you're trying to, I don't know, like stuff it in a box or something. That would be our tertiary structure. So we can see it here. I've got an alpha helix, a couple alpha helixes. They're connected by a salt bridge here, and they're folding in on themselves. So now the alpha helixes, the same alpha helix is able to cross interact. And the way that that's happening is by these R groups. So we fold the protein into a 3-dimensional shape. There's a couple different ways that we can get these tertiary structures. And again, you do want to know these. So we can have hydrophobic interactions. This is between 2 nonpolar amino acids. So if we have 2 amino acids on our chain that are both nonpolar, they will sort of resist or they won't like water. We can have hydrophilic interactions. This is between aqueous environments and polar amino acids. So if we happen to have some polar amino acids on there, they can have an interaction with the surrounding water that will give it some sort of tertiary shape. Again, these get very complicated, right? Depending on what the protein is, what the sequencing is. You don't need to go into that depth, but you do need to know that these are a type of tertiary structure and what the differences are between them. We can have these things called salt bridges. This is an ionic bond between ionized R groups of a basic and an acidic amino acid. So there can be 2 amino acids, one being sort of acidic, one being sort of basic, and they'll kind of stick to each other. And when they stick to each other, you get this bending in the cord. So think about your phone cord. Let's say you have a piece of gum on one end and you have a piece of sticky tack on the other, and those happen to come near each other, right? They'll attract each other and they'll fold the cord. There's also hydrogen bonding happening on our tertiary structure as well. So between the H of a polar R group and the O of an N or another amino acid. And this last one, it's not highlighted, I don't know why, but we can have these disulfide bonds. This is where we have sulfur and sulfur interacting. So insulin, if I go back here, just gonna scroll up real quick. Where were you, insulin? There you are. Oh, come on. Where did you go? we are. Okay. So we've got these chained together, forming a primary structure with our peptide bond. I am not sure if insulin is an alpha helix or a beta pleated. Sorry, don't know, but there's some sort of secondary structure going on. And then this sulfur-sulfur is a tertiary. It's attaching the two together. So that's sulfide-sulfide or disulfide bond. So you're not gonna be tested on this, but this is kind of interesting, just for your information. So sickle cell anemia. Sickle cell anemia is caused by this abnormality in the shape of one of these hemoglobin proteins. So a normal hemoglobin chain has a glue or a polar amino acid right here. If you have sickle cell on this chain, you end up, the glue amino acid is replaced with a val, and this VAL is a non-polar amino acid. That has a huge effect when this thing is polar versus this thing is non-polar. So the 6 amino acid is now non-polar. The non-polar R group is attracted to other non-polar regions along this chain. So this will start to stick to other parts of the chain. Because it sticks to other parts of the chain, it causes these red blood cells to sort of curl or round on themselves, forming this crescent shape. So here is a normal blood cell. Here's a blood cell that's starting to have sickle cell. Here's a blood cell that has pretty dramatic sickle cell. It's because, again, this replacement here is sticking to other parts of the chain, which is giving it this curved shape. Now, this curved shape is not good for blood cells. When blood cells are round like this, it's really easy for them to flow and bounce off of each other. When they have this curved shape, they can kind of clump up on themselves, which can cause clogged capillaries, inflammation, pain. It causes lower transportation of oxygen. So that's an example of where just a little flip, right? Just a tiny little flip on that protein has a huge effect. So we want to indicate whether these things indicate the type of protein structure, whether it's primary, beta pleated, alpha, triple helix. So a polypeptide chain held together, held side by H bonds. So this would be side by side is going to give us this, this beta pleated sheet. Sequence of amino acids in a polypeptide chain. So anytime we're talking about amino acids just connected to each other, that's the pearls on a string, that's a primary structure. Corkscrew shape with H bonds. So the corkscrew is our alpha helix, and 3 peptide bonds woven together, that's our triple helix. So this is what I'm kind of talking about, where this is a little bit more definition based. Your lab gives you some practice on these. Okay, this says select the type of tertiary interaction. This one I'm not gonna have you get into because I don't expect you to have the amino acids memorized. So we are. So one tertiary structure we can have can produce what's known as a globular protein. Sounds very scientific, globular. But they form these globs, or they're compact sort of spherical shapes, so that telephone cord gets balled up into a really tiny compact ball. These things do a lot with synthesis and also transportation. They're good at transporting oxygen in muscles. So the folding in and the riveting of this structure gives us this tertiary structure. This is myoglobin and hemoglobin, so blood cells, things that are in blood cells to transport oxygen. Depending on what your tertiary structure is, some structures will form more fibrous proteins. So when you get these alpha helixes, these a lot of times will produce, this is keratin, it'll produce things like longer proteins, so hair, nails, skin, things that have a stretch to them. you've heard of, so we have alpha keratin. If you've heard of beta keratin, which is in feathers, this will have a beta pleated sheet actually. So if it isn't enough that we have 3 structures to layer on top of each other, we do have a 4th structure, the quaternary structure. The quaternary structure is just the combination of 2 or more protein units. Consists of 4 polypeptide chains as subunits in a sort of hemoglobin blob. Is stabilized by the same interactions found in tertiary. So anything that you found in tertiary structures, all these things, the disulfide bonds, the ionic, the hydrophobic, hydrophilic, all those apply here. What's happening in this quaternary structure? Think about taking your phone cord, okay, that's curled, and you fold it up into a ball, and then you put it in a box with 4 other cords or 3 other cords. That's what's happening at the quaternary structure, okay? These are separate proteins that are kind of globbing or sticking together. So here this shows us the whole setup. So we've got our primary structure, which is the single amino acids that are getting stuck together in a chain by polypeptide bonds. That chain, long chains are not very efficient for storage. So it's much more efficient to curl them up or pleat them. So our secondary structure is going to be alpha helixes, or beta pleated or triple helix. Then we can take that more efficiently coiled or folded up chain and bend it in on itself. If we bend it in on itself, that's the tertiary structure. That's all the alpha helix, or I'm sorry, that's all the ionic, disulfide, hydrophobic, hydrophilic, hydrogen bonding connections. And then we can take that folded up phone cord and we can stick it in a box with some other phone cords that are different from it to make this quaternary structure. So be able to separate those things out definition wise. What are the driving factors causing it, right? So this is a peptide bond, whereas this is hydrophobic, hydrophilic. Here's a bunch of information again to sort of summarize all that stuff. Let's try a couple of these. So I want you to tell me whether these are describing a primary, a secondary, a tertiary, or quaternary structure of a protein. I'll give you just a minute. Okay, so for A, it says beta pleated sheet. So that is an example of a secondary protein structure. It's where we have hydrogen bonding, taking our primary chain and folding it in on itself. Order of amino acids in a protein. That's the simplest thing that we can do. How are those pearls lined up on that string? That would be a primary structure. C, a protein with 2 or more peptide chains. So anytime you have 2 different peptide chains clumped together in a box, that is a quaternary structure, or your 4th. The shape of a globular protein. So what determines this shape would be a tertiary structure. So how is it folded in on itself? And then disulfide bonds between R groups. This is something that we would see in a tertiary structure. We could also see that in a quaternary structure, but really that's more tertiary. So there's a lot of things going on to make proteins, right? Something as small as changing out the ordering of 2 of the pearls on your hemoglobin chain can give you sickle cell anemia. If some of these secondary or tertiary structures start to fall apart, like when our secondary triple helix falls apart in our collagen, our skin starts to sag. We can't regenerate as fast. How do these proteins start to fall apart? Well, there's a couple different ways that we can it's called denaturalization of proteins, or we can disrupt bonds on the secondary, tertiary, or quaternary level to mess with the effectiveness of the proteins. So denaturization of protein can happen through heat and also organic compounds. These will break apart any sort of H bonds and disrupt hydrophobic interactions. If we have a protein and we put it in an acid or a base, that can also break apart H bonds between polar R groups, and it can also break up or disrupt these ionic bonds. Heavy metals. Heavy metals tend to react with our sulfur-sulfur disulfide bonds, making those break apart. And then agitation, that's a mechanical thing. So like whipping something, that will stretch the peptide chains out until they break. So let's look at some of these examples of denaturization. Why does an egg look different before and after you cook it? Right, well, what happens in this is that we heat up the egg and it breaks apart the bonds in the tertiary structure. So that crumpled up coil now sort of elongates itself. It's one reason why you can't like make the egg go back. You can't make a cooked egg back into an uncooked egg because you can't reconnect that protein. Some ways that denaturization can happen. So situations where we are above 50 degrees Celsius, so cooking food, and then, but this is important, if we can denaturize bacterial protein, we can also use it for autoclaving or sterilizing stuff. So when they heat up equipment, either in a medical setting or in like a spa setting, they're denaturizing the bacteria's protein. Acids and bases are also a way. So the way they disrupt is the hydrogen bonding between our polar R groups, and they can destroy salt bridges. So examples of this: lactic acid from bacteria, which denatures milk protein to form yogurt and cheese. Organic compounds can mess with our hydrophobic interactions. So ethanol and isopropyl alcohol can help to disinfect wounds and prepare the skin for any sort of injections. So again, we're using this organic material, ethanol, isopropyl alcohol, to disinfect, to break apart bacteria's protein so that it dies. Heavy metals can mess with disulfide bonds. If you've heard of mercury and or lead poisoning, that's happening. Why do you get sick from eating heavy metals or having lots of heavy metals? Because they're literally breaking apart your proteins on like a microscopic level. Not good stuff. And then agitation. One way that we can agitate, like you can take egg whites and you can whip them to form a meringue, or whipped cream when we whip heavy cream into yummy whipped cream. We are stretching out these polypeptide chains. We're breaking some of the hydrogen hydrophilic bonds, or hydrophobic bonds rather. Okay. So the last thing I want to talk about for the semester is enzymes. So what is an enzyme? An enzyme is a type of protein. Okay, so it's got its own protein chain and its own primary, secondary, tertiary, and quaternary structures. But what an enzyme is, it's not just some boring old protein. It's a protein that actually acts as a biological catalyst. So remember, a catalyst is something that lowers the activation energy of a chemical reaction. So it makes it easier for a chemical reaction to occur. We have enzymes all over the place in our body. On the surface of an enzyme, so here's our enzyme, there's what's known as an activation site. And the activation site will bind a substrate, and then it will, so the substrate will come in, hit the activation site, the enzyme will do its work, and it'll break this substrate apart into products. So let's look at that in more detail. So again, enzymes are catalysts. They are catalyzing reactions in our body. So if we look at this here, we've got CO2 and we're combining it with water to produce HCO3 or bicarbonate and H+. In order for this reaction to occur, because otherwise, if we didn't have this activation energy, reactions would just fall forward always. So we do need to apply a little bit of energy to move up and over the activation energy so that we can then move towards the products. Without an enzyme, we have a really high activation energy. With an enzyme, it makes the reaction much quicker. So it'll increase the rate of reaction, or it lowers the activation energy. How do we get the name of an enzyme? Hold on one second. Sorry, I had to answer something. So the name of an enzyme is derived by replacing the end name of the reaction or reacting compound with ASE. So sucrase is catalyzed the reaction of sucrose. Oxidase catalyzes an oxidation reaction, so on and so on. I'm not really going to test you on the names of enzymes. But we are going to talk about this. So what an enzyme is actually doing. So on our enzyme, this is a generic enzyme, we have this thing called an activation site. And what is the activation site? It's a region within an enzyme that fits the shape of whatever molecule it's trying to react to. And the reacting molecule is the substrate. So let's say we're given glucoses in our system. We would have an enzyme that helps to break down that glucose into what we need. It contains amino acid R groups that bind to the substrate, releases the product when the reaction is complete. So this activation site will take the substrate, it'll hold on to it, it'll do its thing, and then it releases the substrate and we will get products. Now, the interesting thing about a catalyst is that it helps to speed up a reaction, but it itself is not consumed or changed. So the enzyme stays the exact same. It helps to run the reaction forward, but it doesn't change. So you can use it over and over and over again. So this is sort of a simple reaction that happens, this enzyme catalyst reaction. So a substrate, here's our substrate S. It attaches to the activation site on the enzyme. So I have my enzyme, I have my substrate. They're separated by a plus, they're not combined yet. And what will happen is they will form this thing called an enzyme substrate complex, or the two of them are sort of fused together temporarily. After this happens, the enzyme releases the products, and the enzyme stays the same thing, right? It was a blue E here, it was a blue E here, it's a blue E here. What happened to the substrate? It bonded and then it became a product. So the substrate changes to a product, but the enzyme stays the same. So in the hydrolysis of the disaccharide sucrose, the ES complex, or the enzyme substrate complex, is formed as sucrose binds to the activation site, specifically of sucrase. So sucrase is the enzyme, right? It's doing its work on sucrose, so we ASE it. What happens is the glycosidic bond in sucrose is hydrolyzed. So we break that bond in the sucrose and it breaks it down to glucose and fructose. So our body will break the sucrose into the glucose and fructose, and then our body can use the glucose and fructose as needed. The enzyme, though, stays the exact same. So we can think about the enzyme substrate in a couple different models. The first model is what's known as the lock and key model. This is where the activation site is very rigid and non-flexible in shape. So for instance, sucrase, it is this very, very rigid enzyme and it will only plug in the sucrose, and the sucrose has to have sort of a perfect fit or a lock and key fit. The substrate is the key that fits to the lock. Now that isn't necessarily what's happening. Really, we have more what's known as an induced fit model. So the enzyme structure is more flexible. So it's not so rigid, and it can adjust its shape of the activation site in order to bind to the substrate. So if it's really, really rigid, like this lock and key, you know, God forbid anything is off on that glucose, or rather the sucrose molecule, it's not going to fit. But if we have this induced fit, the enzyme can adjust a little bit to maybe fit a sucrose that is like a little bit too big or a little too small, or something is slightly off, and it can still use it. So the range of substrate specificity increases. This sort of induced fit increases its catalyst capacity. It's better for reactions. So here in the induced fit model, we've got our activation site, which is, you know, it's sort of like this little boat shape. And we've got our substrate, which is kind of like a circle. What happens is this will kind of adjust its shape slightly to bond these two things together. And then it will release the product and it returns back to its shape, its original shape. So it's got this flexibility to it. Actually, I'll answer a final question. Someone asked me about the final date. We'll talk about that after we finish lecturing. Let's get through this and then we'll do it. So, let's see. I'm not going to talk about this. So the activation site is A, the enzyme, B, a section on the enzyme, C, the substrate. The activation site is B, a section along the enzyme. In the induced fit model, the shape of the enzyme when substrate binds, B, adapts to fit the shape of the substrate. We're going to keep that pretty simple. There are things that can affect enzyme activity. So the activity of an enzyme describes how fast an enzyme catalyst or catalyzes the reaction. How good can an enzyme convert a substrate to its product? It's affected by a couple different things, including temperature, pH, and the presence of what's known as inhibitors. So as far as an enzyme activity and temperature, sorry, it keeps flipping on me. Enzymes are most optimally active at like 37 degrees Celsius in human beings. If it gets colder, the enzyme doesn't work as well. Like lower temperatures, you know, things get cold. When things get cold, what do? You kind of curl in on yourself. You're not as active. So same thing happens with enzymes. So we heat it up and it gets to this like happy place, you know, maybe at, I don't know, like in South Carolina or Florida on vacation, right? It's nice and warm and you're in a good place. But if you're in South Carolina in the middle of summer, you've denaturized, okay? Because it's so hot there that you start to break apart. The same thing that will happen with the enzymes. So they will denaturize after you increase, you know, past 50 degrees C. As far as pH is concerned, enzymes are most active at an optimal pH, contain R groups of amino acids with proper charges at optimum pH, lose activity in low or high pH. So the optimum pH really is going to be dependent on what the enzyme is, right? If it's an enzyme that's like in your stomach, it's going to have a different optimal pH than something that's in your bloodstream. So we have that. Happy pH, here are some of the different optimal pHs for the different things. So like pepsin, which is located in the stomach, an enzyme in the stomach, it's optimum. It's really happy at a very, very low pH. Why is it happy at a low pH? Because it's living in your stomach and that's all it gets. So it's had to adjust to be happy with that. Arginase, which is in the liver, is much happier in a basic situation. Your liver has got a lot of basic material in it. So if, you know, this ended up in your stomach, it wouldn't work as an enzyme. Sucrase, which is an enzyme that breaks up sucrose, has an optimum temperature of 37 degrees Celsius and an optimum pH at 6.2. It says determine the impact on the enzyme's activity. So changing the pH to 4, if it likes 6.2 and it changes it to 4, we're going to say that that's definitely going to decrease it. Running the reaction at 70 degrees Celsius, that's going to denaturize it. So it's going to decrease it as well. So both of those will decrease. We also have what's known as an inhibitor. Inhibitors are molecules that cause a loss of catalyst activity. They prevent the substrate from fitting into the activation site. So the catalyst is trying to do its job. It's going along. It's trying to convert the substrate into a product. And then all of a sudden, this really tempting inhibitor shows up and it like grabs onto the inhibitor. There's a couple different types of inhibitors, but this one is showing a competitive inhibitor. So the enzyme is like, ooh, look at that. I'm going to bond to that. Well, while it's bonded to that, it's no longer doing its job with the substrate. So a competitive inhibitor has a structure that's really similar to the substrate, and it competes for those activation sites. Has its effect reversed by increasing the substrate concentration. So let's say your body's like, oh, there's an inhibitor there. Well, I'm just going to add more substrate concentration. Well, you'll go back to having a more balanced situation. So here's our enzyme. There's the substrate. It's going to bond with the substrate and it's going to get a product. Well, if this enzyme instead gets distracted by this shiny inhibitor over here, it's now bonded to the inhibitor. This substrate is not able to bond. It may bond with another one, but it's going to slow the whole process down. We also have what's known as a non-competitive inhibitor. So a non-competitive inhibitor, it doesn't compete, obviously, but what it'll do is it'll bond or it'll attach to some other place outside of the activation site, and it like distorts the shape of the enzyme. So it prevents the substrate from actually being able to fit into the activation site. This is important in that it can't have its effect reversed by adding more substrate. So if I put more of the substrate in, it doesn't matter, it doesn't fit, right? So here's my normal situation, my enzyme combines with my substrate, and then it produces the product. Here, my enzyme gets like smushed. It bonds with this somewhere else and it changes this shape, and this substrate can't fit in there, so we can't get the products. We also have irreversible inhibitors. So in an irreversible inhibitor, this isn't a temporary thing like this guy. This guy is temporarily fitting in the activation site. This one is temporarily adjusting the shape of the enzyme. In an irreversible inhibitor, it's a molecule that causes the enzyme to just literally lose its activity. It doesn't work anymore. We find this a lot in toxic substances. They'll just destroy the enzyme. They'll form a lot of times by covalent bonding with an amino acid side chain. So it destroys it. Not good. So here are some of the different types of inhibitors, sort of just summarizes everything that we talked about. So let's identify each description as an inhibitor that is competitive or non-competitive. So increasing substrate will reverse inhibition. Well, the only thing that can have that happen is a competitive situation, right? If I add more substrate, there's more competition. It binds to the enzyme surface, but not the activation site. That is a non-competitive, right? That's what our non-competitive, where it adjusts the shape. Its structure is similar to that of the substrate. That's a competitive situation. And then inhibition is not reversed by adding more substrate. That's a non-competitive. So you guys seeing how this last chapter and really these last two chapters are kind of more definition based? Expect to see that on the test. All right. Oh, that was it. Ooh. The semester's over. Kind of uneventful, huh? So that was the last of it. So a couple things. I'm going to keep sharing my screen with you because I want to go over what the last little bit looks like here. So we are almost done. And this is really the last formal week of classes. Next week is finals week. Let's see what we got here. So your fifth test will be this Sunday, Monday. You'll cover just chapters 13 and 16. Somewhere in the syllabus, it did talk about, like, if we had time, we were going to do nucleic acids. We're not going to go over nucleic acids, so you don't have to worry about that. Your test will be, your final will be on the 16th and the 17th. So you can take it from the 16th to the 17th, which I believe is Thursday, Friday. I believe I gave you the longest amount of time to study. Pull up my calendar. No, it looks like it's Wednesday, Thursday. All right, well, we'll go with Wednesday, Thursday then. If for some reason that doesn't work for you, just message me and I'll try and I could let you take it earlier. I couldn't let you take it later. We don't have class on Monday, but I am around and available. So if you want to do like review and stuff like that, that's fine. You just got to message me and then we can show up to normal lecture setting. If you have any last minute labs or homework or anything, you want to try and get some partial credit, make sure you get that to me by Sunday. Otherwise, we're done. What else do you want from me? You guys need anything from me for today? If you want to review, if there's anything you want to review from anything in the semester, I can throw up some example problems. If you want to wait and do some review on Monday, that's fine. You just got to message me to let me know to show up, and then I'll show up. So just to check, we have 3 labs and 2 mastering chemistries due this Sunday. That is, I believe that is correct, yeah. Are you gonna go over the enzyme lab? Can we do a review on Monday? How do you do number 11 and 12 in the dry lab for the amino acids? Okay, so questions, great. We can do a review on Monday. Yes, I'm totally fine with that. I have nothing going on. Haven't had anything going on for 9 months. All right, let's pull out let's do the amino acids dry lab. Let's look at that, yeah. If you're wondering where the amino acids dry lab is, I did insert it in the Moodle page, so you can find it down here. It says fill this out, you'll submit it like you normally do. So give the 7 types of proteins and their functions, definition really. Draw the general structure of an amino acid, circle and name the 2 functional groups found in the structure. So this is just drawing it. The aspirin lab. No, we're not doing the aspirin lab. We're not going to do the aspirin lab. I don't even think I have a submission for the aspirin lab, do I? Do I? I do? No, we don't have to do the aspirin lab. Don't do the aspirin lab. I don't want to grade it. I don't want to grade like 65 million labs this weekend. So you have to turn in the carbohydrates lab and you have to turn in the, where am I? The amino acids lab. I'm doing advising right now as well, so my brain is like in 1,000 different places. Define essential amino acid. How many amino acids are there to build? Okay, so these are all just definition based. Let's look at specifically, what did you say? "11 and 12. If you already did it, can you get extra credit? Sure, why not? Throw it on there. My assumption is the people that have already done it probably don't need the extra credit, but go for it. You can turn it in. You overachievers, yep. So for these, this is talking about the pI. So like if you're at your optimal pH. So remember, if you're below your optimal pH, you will add hydrogen. If you're above your optimal pH, you'll take away hydrogen. Specifically, you'll add hydrogen to the COO group, or you'll take away hydrogen from the NH3 group. So if you can believe it, I don't have my amino acids memorized, so I need to look them up. Because who has the brain capacity to memorize all that stuff? Somebody does, just not me. All right, I'm gonna pull my amino acids up here. So this first one is alanine. And I want to know what its pI is. Okay, I'm going to go over here. So for those, you're going to have to look some stuff up. Look up what the symbol or the molecule shape is, and then look up what its pI is. So for alanine, whoa. It has an optimal pH of 6. And we said we're in a situation we're actually at a pH of 2. So I'm going to draw it if it's at a pH of 6. So we've got our C, It's bonded over double bonded. Nope. This is my alpha carbon single bonded to a carbon that's double bonded to an oxygen and then bonded to another oxygen that has a negative. Then this alpha carbon has another group coming off over here. It's got the N with the 3 hydrogens. And this has got a plus charge. And then it also has a hydrogen coming off of here, okay? So this is sort of the base of every amino acid. Then it's got an R group coming off of it. That's what changes. So what is the R group for alanine? It's a methyl group. So I'm going to take that R and I'm just going to replace it with the methyl group. But nothing's really going to change, CH3, to this when we change pH. Okay. If I go to a pH of 2, what is happening here? Are we adding H plus or are we subtracting H plus? We are adding H plus because it's acidic. So where on this structure can I stick an H? The only place I can stick an H is right here. So my structure, if I'm in an acidic solution or more acidic than my PI, this is, I don't just want to call it pH, I want to call it PI. It's optimal pH, is going to look very similar to this structure, an alpha carbon bonded to a carbon over here, double bonded to an O, single bonded to an O that now has an H. This is the magic addition. What happens to the charge on this side? It's no longer negative, it becomes neutral. Okay. This over here stays the same. N, H, 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 this has an H, and this also has that CH3. So the only thing that's different is what's happening right here. This is also a plus. What happens to the overall charge of this thing? Charge is now positive. Here it's got a positive and a negative, so those are neutralizing each other, neutral pH, or I'm sorry, neutral charge at its PI. If your pH is lower than your PI, your charge will become a positive because you've packed in a hydrogen. Why don't we just for fun go in the other direction? So let's say instead we're going to a pH of 11. In this case, we're removing H plus. Well, looking at my structure here, where can I take hydrogen? I'm not going to take this hydrogen. I'm not going to take this hydrogen. It's going to happen off of one of these functional groups, specifically here. So now I've got my carbon. It's bonded to another carbon that's double bonded to an oxygen that's bonded to an oxygen that has a negative. The alpha carbon has a hydrogen down here. And now it's bonded to this N that has an H and an H, and it's down an H. So it's lost this H, removing the H. So what happens to the overall charge here? It went from positive to neutral. So now there's a negative side to this molecule. Let me add the R group, CH3. Okay. So the charge here is negative. Now, it doesn't matter how complex this group gets. That's not where we're playing around with pH. It's all happening at this base of the amino acid. So when looking at that worksheet, when it says, you know, aspartic acid, you just need to look up what is the PI for aspartic acid and then draw the structure. I mean, the only thing that changes is the R group. Everything else will play around the same, all right? This says if we put it above 7, so you need to figure out the structure for valine, the structure for serine. And again, all of these you can find in the slides. We go back up here. Here are a bunch of them. Okay, there's valine. Here's serine, right? And it also has its isoelectric or its PI points. So serine likes to be at 5.7. So if we're at 7, we're in a more basic solution. What was the other one? Valnine likes to be at 6. So again, we're in a more basic solution. So you'll just play around from there. Does that help clear that up? Yes. All right. Cool. So hopefully not as scary as it initially looked. Then you just got some definitions. This is a pretty basic lab. It's not too bad. The other, so aspirin lab, you can do for extra credit. We talked about how to do that. We can review on Monday. We went over the enzyme lab. Okay, other questions? If not, we can finish for today, and then I will just be, I'll be available on Monday. I'll log in on Monday like I normally would, so that we can just have a review. Come with questions though, okay? So that it's not just me sitting here staring at the screen and you staring at the screen and nobody talking. Awesome. If you have any personal questions, feel free to stay around. Otherwise, congratulations, you're almost there. Good job. Woo-hoo. Nice work. If I don't see some of your faces, it's been really great having you in class. No, it's been online. It's super weird. Message me if you have any issues on taking the test, taking the final, anything like that. All right.